I'm also very happy to welcoming everyone to our Cardiac Safety Symposium 2025. We will be having great speakers today, highly interesting topics, basically all embracing next generation toolboxes for predictive drug development and cardiac safety. So to start with, let's have a look at what is happening, what has happened already in 2025 in the cardiac safety world, and what will be happening in this year. So the ICH E14 S7B implementation working group, for example, announced that the new guidelines are expected to reach the final step 4 adoption stage in November this year. This includes the adoption of the second stage S7B Q&As and the updated E14 Q&As by the regulatory members of the ICH assembly. And now with the anticipated completion of S7B Q&As provides a strategic opportunity to integrate the outcomes also into S7A, because strong momentum is building here at the moment, especially Derek Leishman and Jean-Pierre Valentin advocate for a comprehensive revision of the ICH S7A guideline to address gaps in safety and secondary pharmacology assessments. So what else happened this year? Well, on April 10th, the FDA announced a major policy shift to phase out animal testing requirements for monoclonal antibodies and other drugs. So this move is designed to improve drug safety, accelerate drug development and platform and cell development, reduce costs while addressing ethical concerns about animal use. Now, the roadmap basically foresees to reduce, refine, and replace the traditional animal models by a range of different approaches, the new approach methodologies, NUMS. So, for example, 2D models in vitro systems, or 3D cell models like spheroids or organoids, or organs on a chip, like the liver chip is mentioned in the FDA roadmap. And all of this in combination with computational models. And this is actually something that we will be hearing from Stuart, which is our first speaker, and he will have some great insights there today. So basically, the recognition of the new approach methodologies, being able to deliver a robust science was a great achievement by this roadmap. And here actually a practical and phased rollout is described. So the implementation strategy is incremental. It all begins with monoclonal antibodies. And of course, a long-term adoption across drug classes is planned. Furthermore, in the roadmap, there's described the case-specific waivers, the promotion of data sharing, and validation pilot programs. Now, by end of 2030, the animal studies should be indeed the exception rather than the norm. And instead, comprehensive toolboxes like human cell models plus computational models shall be the new standard. Well, and this is indeed what a lot of cardiac researchers have been waiting for. So this is why a lot of things that you guys are doing, why we are all doing what we do. For example, Nanyon is scaling up and developing these in vitro systems. And it's also why we are actually meeting here today in such a cardiac safety symposium. So to unlock functional human data that informs safer and more predictive drug development. And indeed, we are all together having success. So we will be listening to some great comprehensive studies today. And of course, a lot happened already in the past. And before we jump into the presentations, I would just like to show you some brief examples of some achievements. So for example, matured stem cell-derived cardiomyocytes here in a toolbox with about 2.5D environment on a flexible membrane. These cardiomyocytes show positive inotropic effect towards compounds, for example, isoproterenol. This means mechanical environment promotes mechanosensitive regulation on the cellular level. There is another example showing translatability, the positive inotropic effect towards omecamtiv So this concentration-dependent effect and increased duration of the signal is just as reported in vivo studies. There's another example, iPS cardiomyocytes in co-culture, here with cardiac fibroblasts. So again, this co-culture here is boosting the situation. So you can see that the co-culture shows an increased contractile force of the cells. So basically, a co-culture system better replicates the complexity of native tissues, leading to a more physiologically relevant behavior of the cells being investigated. I'm bringing this example here also because the FDA did mention the liver chip in their roadmap. So there are stem cell-derived hepatocytes out there showing characteristic functions and morphology, for example, here shown in an imaging system where we have stained cells showing the existence of the cell-cell contacts, which are characteristic for the hepatocytes. And this was also proven in an impedance system where the development of the transepithelial electrical resistance was monitored over the days. And indeed, the metabolically active iPSC-derived hepatocytes demonstrated a time and dose-dependent toxicity, for example, upon aflatoxin treatment, which is very nice, I think. And another example, so 3D cell constructs is a very important topic, like spheroids or organoids. In this case, the researchers were using HD29 spheroids, so these are cancer cells. Here you see two of the spheroids sitting on an impedance measurement electrode. And you can nicely monitor the adherence and a typical reorganization of the cells sitting in the new matrix. And then a proliferation of the cells, the spheroids getting bigger, and then in the end being killed. And now my last example, atrial and ventricular stem cell-derived cardiomyocytes, so chamber-specific cells. These cells were investigated in a high-throughput automated patch clamp system, looking at the effects of compounds like carbachol to ion channel currents. And a similar type of cells has been investigated, again, in a contractility measurement system, again, looking at chamber-specific effects, So carbachol reacting on atrial but not on ventricular cells.